Good evening, participants in the fifth day development certificate course in data analysis. And our today's speaker is Dr. Ankur Bhardwaj. It is my pleasure to introduce our esteemed speaker for today's session in this skill development certificate course on data analysis, Dr. Ankur Bhardwaj. Dr. Bhardwaj currently serves as an assistant professor in the Department of Botany at Ramjas College, University of Delhi. His academic journey is marked by excellence, having earned both his MPhil and PhD degrees from the prestigious Department of Botany, University of Delhi. With over a decade of teaching experience under his belt, Dr. Bhardwaj is a specialist in the fascinating and evolving field of genomics, bioinformatics, and biostatistics. His commitment to advancing knowledge is evident in his extensive publication, which include numerous research papers in reputable national and international journals, as well as contribution to book chapter in plant genomics and statistical biology. Dr. Bhardwaj has also successfully led three research projects supported by recognized bodies, showcasing his ability to translate theoretical knowledge into impactful practical application. His expertise extends a published book on biostatistical analysis, further solidifying his contribution to the field. Beyond academia, Dr. Bhardwaj influenced REACH national education initiative as he has served as a subject expert with NCERT on multiple occasions, contributing to the development of their curriculum. Currently, Dr. Bhardwaj research endeavor focus on the exciting intersection of molecular data and statistical insight, fostering.
Yeah, thank you so much, Dr. Savin, for this introduction. I am, first of all, thank you of Zakir Hussain Delhi College for giving the faculty this opportunity to organize such workshop, and especially Dr. Savindra and Dr. Zishanur Rahman, who contacted me to deliver a lecture in this ongoing series. And it would be a good experience for me, and I think it would be a great opportunity for students also to be in this these lecture series. And I would like to mention that I think Dr. Sabin has mentioned one book which I have authored. He is also one of the author in the book. So, yeah. So, can I start with the lecture here? Yeah. Shall I start with the lecture?
Yes, sir, you can start.
Yeah. So, first of all, share my screen. One thing I just remembered my teaching career with Zakir Hussain College only. I joined as a guest for some time, and it was my first teaching profession, you can say.
In year, sir, which year?
A long time back.
I think before I joined that college.
Yes, you were not there.
Okay.
So let me quickly share the screen. So is the presentation visible to the candidates?
Yes, sir. It is.
Okay. Thank you. Friends, it's I'm sure that all other persons who have given talks in this series, they have also mentioned that biostatistics or the statistics is a field that is used in all type of research these days, whether it is molecular biology or it is some other field like genetics, ecology, cytogenetics, any other thing. Right. So that is why it becomes for the students to be very proficient in this field. Just give me a moment. I will just go with the settings of my PC. Yeah, right. As I was saying that this is the
Can you
field.
share it in presentation mode, sir?
I will be using a draw tool also. So
Okay, okay, okay,
that requires me to open it in this format only. When I go in the presentation mode, it stops working.
okay, okay.
So I think it will be fine to students to
Yeah,
see.
sir, it's visible. And sir, one clarification that there are participants from various backgrounds, like there are undergraduate,
Right,
postgraduate, research scholars, scientists, and many faculties.
right, right, right. So, considering this only, I have kept this presentation quite simple, and I will be covering mainly the conceptual things only. Like my point is to make the concept more clear, because as soon as it comes to statistics or biostatistics, students think that they have to do a lot of numericals. So numericals or tools are also part of this, but having a basic understanding is the first thing that is required. So keeping in this view that the students might not have mathematics at their class 12, students from other backgrounds might have joined. So I have kept the simple quite simple, and that is why I have kept this title Introduction to Measures of Central Tendency. So let me proceed with this. All right. So as this series is going on quite a few lectures also, so many of the uh, professors and resource persons who have been here, they must have talked about the origin of statistics and what is it all about. So we say that it starts with a way around 17th century, mid 17th or later 17th century, where people used to collect data and about the person's health, wealth, their resources, and this data is used by the state or the government, and that's how this term started originating. In German, it is called statistic or the status word uh, in Latin, and later on, it's just got modified to statistics. Right. And obviously, the roots of this branch of science is in mathematics. Right. So, mathematics is the branch of science that gave birth to statistics. So, is there anything that mathematics was not able to solve? Mathematics is a very robust field, right? Mathematics is a field which have produced great scientists. So, is there anything that mathematics was not able to solve? So, and that is why a new branch originated within the roots of mathematics called statistics. And whenever we use statistics with biological data or in context of biology, we start calling it as biostatistics. So, is there can any student answer this question? Whatever you comes in your mind that if mathematics was also there, so why statistics came?
Because statistics can also work on qualitative data.
Yeah, but like mathematics can also find that the topics that I'm going to cover, like mean and the median and the mode, they are also there in mathematics, right? So what's the difference? So let's say
Actually, statistics mainly gives the meaningful conclusion of any particular data
so
kind
so
of thing.
so
So
in this way, are you saying that mathematics was not able to give answers
no,
for a
it's
particular
giving,
question?
but like how to interpret the data in a meaningful conclusion and kind of like analyzation, interpretation, and in that manner, I think it more informative rather than.
So mathematics was not able to do this.
नहीं नहीं no sir, it can be, but
Let's
what
understand this.
the,
Let's
yeah,
understand this with one more example. This will make you more clear. Let's say there is a girl in your college whose name is Neha, right? So if I
okay.
ask her that how many girls are there with this name in your college, so out, but she will take a lot of time, right? She has to go to every class, make a list of all the names, and then she can find out that these many girls have this name, right? And if I extend this question that how many girls have this name Neha in the entire Delhi University, right? So now it looks like impossible for her. But with the help of some volunteers, like 100, 200 students with her, they can also manage to do this, but it will take a lot of time, right? So this is the task of mathematics, that mathematics gives very concise, particular answers, right? But when it comes to very large data, the approach of mathematics is taking a lot of time and it is taking a lot of resources. And that time statisticians came, they say that if you want to find out that how many girls in the entire Delhi University, or let's say in the entire country, you want to find out how many girls have this name, we can give you this answer with quite certainty, with a very less effort. What we will do, we will take some samples from different states, from different universities, and based on this, we will find out this. So that's these were the some questions where statistics had an edge to take less time, less resources, right? And it started also using computational tools, which made it better. Now coming to today's thing. I think this is that we say that the statistics is broadly of two types. One is called the descriptive statistics, and the other one is the inferential statistics. Right. So the descriptive statistics is the statistics where we summarizes the data and we describe the data. Whenever you have some data, so this data will have some properties, and descriptive statistics talks about these properties. Right. Descriptive statistics includes this measures of central tendency and it includes measures of dispersion. Right. So I will be talking about these two today. In the central tendency, we have three measures called mean, median, and mode. And in the dispersion section, we have range, standard deviation, and variation. Quartile deviation is also there. Right. And this is the inferential statistics is more valuable and I can say an essential extension of statistics. So it gives you conclusions about future predictions. Let's say there is one student who came first in class 1 and in class 2 also, class 3 also, class 4 also, 5, 6, 7, 8, 9. In all these classes, he stood first, or let's say he scored 60% of marks. So will he score a first division or more than 60% marks in class 10th also? So descriptive statistics cannot give you this answer. Why so? Because this event has not yet happened. The student has yet to give answer and the result has yet to come. It can only tell you about the property of the last 9 years data. It can help you understand this last 9 year data, but it cannot jump into any inferences about the event not yet which is not yet happened. So this thing is taken care by inferential statistics. Talk of today will focus on this part, which is called descriptive statistics. So before going into the details, let's understand what we mean when say that when we have a data or data in plural. Data is singular and data is plural. So any observation that we have, any observation where we have raw facts or we have collected some information from different sources that is recorded becomes part of data, and this data will be later on analyzed. So it can be anything. It can be anything ranging from this is the most common that teachers use or resource persons use: student scores, student's height, student weight, the height of a tree, the width of a tree, the number of any animal in your college, right? So this constitutes some kind of data. Going beyond this, we can have data of weather like temperature, rainfall, humidity, right? And in fact, whenever you take surveys where there are questions which are answered in the format of yes, no, or maybe only, right? So these will also constitute data. And the most important thing about data is that it itself does not have any meaning, right? Until it is processed and interpreted. And that is why statistics is important, where we collect data and we process it through different ways, and later on it is also interpreted to give some significance or meaning. When you collect any data, it is just a collection of facts. It will lie there if you do not process it and if you do not interpret it. And even if others don't understand what you have done, that is also important. You have collected and good data, and it's huge numbers: 10,000, 50,000, 80,000 numbers are there. So you also have to find a way to make others people to understand that what this data is all about, and that is where the visualization of data comes in, right? A person who is not in academics, right, or who is not good with numbers, and or let's say the person does not have much time to go into the details of this data, right? So that person would like to have a visual representation where he can make out that what this data actually means. There are different ways like bar graphs, pie charts, line diagrams, histograms, scatter plots, and other things also, which gives visualization to data. So now this data, I am particularly talking about only numerical data. The data can be of different types. For this class, I am considering only numerical data, and this numerical data is the most easiest data, I will say, to understand. It is also most easiest to make students learn about the statistical things. But whenever we have this data, this needs to be first categorized into a particular category, right? Data can be categorized as an individual series, it can be categorized as a discrete series, or it can be categorized as a, and the central tendencies, which I will be talking about later, they can be calculated in this, they can be calculated in this, and they can be calculated on this. Right? Let's first see that how these series look like.
Excuse me, sir.
Yes, yes.
Sir, our participants have already watched they already have these lecture series on different kinds.
Right. So I will move on to the next one. Yes. So in this lecture, I will be talking about examples with individual series only, right? Because it takes a lot of time to have discussions on these two series, right? There are different formulas for these series. I took this context only because when we say we have to calculate mean, when we say we have to study median, or we have to study mode, or any other measure of dispersion, so these can be calculated in these three also, right? Or should be calculated. But for this class or for this lecture, I will be taking examples from individual series only, because this is the most easiest one to understand. Right. So central tendency, the main topic of today, is a central value. It will be the core representation of the data. When you have a data, let's say you have a data of marks of a student in a particular paper, right? So this central tendency will give you a single value that will describe the entire data, right? So what is this helpful? This central tendency, it helps to condense large data into smaller values that can be easily understood by persons. So there are three central tendencies are there when we talk about data. The first one is the mean. Mean, or it is also sometimes referred as the arithmetic mean, right? It is the average of a data set, right? And it is the foundational concept in statistics. So let's say why this data is important. Let's say the admissions are going on in Delhi University this time, and someone asks that which college is better for a particular subject or a particular thing, right? In terms of marks, right? So there are hundreds of students in Zakir Hussain, hundreds of students in Hansraj College, and there are many students in other colleges. So you cannot give them the marks of all the students, that because someone will score less, someone will score more. So the average value will give you a single value that you can use, that the average of the marks in a particular college is this last year. Let's say in Zakir Hussain, the average marks of the student was 80%, and this year it was 82%, and the next year it was 85%. So you can see that the marks are keeps on increasing, and that is going to help the parent that the students are improving every year, right? And this can be used in any type of data. The mean, which is the arithmetic average, how it is calculated, it is the simplest one to calculate, right? It is the sum of all the observations, sum of all the observations divided by the total number of observations. Right. So here mean is represented by x bar, right? You make this symbol x with a bar above it. Right? This is the representation of mean. This is the sign of summation, sum of all the observation divided by n. Now let's take example of this data. It's quite simple. It has four values: 6, 3, 8, and 5. Let's say it is the marks for a particular paper, or the number of trees in a particular locality, right? So simply you have to add these numbers and divide it by all the total number of observations. So total number of observations here is 4. So you take all these numbers, divided it with the total number of observations, and you get the mean. And in fact, the mean, median, and mode is so basic in mathematics that it is, I think, its concept starts from class 6, 7 onwards, right, in schools. So the advantages of using mean is that you can describe the entire data, right, with a single value. Like the principal asks, how is your class doing? Right? You can help with the answer using the average value, a single value that is going to describe the entire data. Right? And one of its main advantages that it is going to consider all the observations. There are measures of, there are parameters which rely on certain values, but the mean takes into consideration the entire data set. So that is a good thing. It is an advantage for this, right? And the other thing is that it is easy to calculate and easy to interpret. The main problem with the mean is the presence of outliers. So here we have a data set where you have to calculate mean from this, right? So you can see that all these values till here, they are quite similar, right? They are quite similar. But there is one value which is very far away from the others. So the far away values will be called outliers. It is on the upper end. You can have outliers on the lower end also. Let's say we have this. So this will also be a outlier. So these outlier will affect your mean. Let's say you have two values, 5 and 5, and you have to calculate the mean, right? And if you have two values, 1 and 9, and you have to calculate the mean, right? So these values are very far away from each other. So in such case, this becomes one of the main disadvantage of the mean. The other thing is that mean works good when you have a normal distribution of data. We will talk about this in, I think, in one or two slides, that whenever you have a normal distribution in a data, then only your mean will be accurate or more near to reality. If your data is skewed or it is not normally distributed, right, your mean is not going to be correct. Here you can see in this graph, we have salary distribution of different employees. So our uniformly or normal distribution data that follows a bell curve will have mean at its top, right? While this is an example of a skewed data, right? The mean will shift from its position and it will not give you the correct answer. The other parameter that is there in central tendency is the median, right? Median, in simple term, we can say it is the middle value, the central value when the data is arranged in ascending or descending order. Whenever you have any data, first you have to arrange it. You can arrange it in ascending order, you can arrange it in descending order. So median will be in the, this, you have data, so it will start from the smallest and go to till the highest. Let's say we have 1 here, we have 100 here. So the median will be in the middle section. This median divides the data into two data sets. So 50% of your values will be below the mean or they are below the median. Whatever median value is there, it will be below this, and the rest remaining 50% will be above it. So the median is going to split your data into two halves. It is less affected by the outliers. This is one of its property, and this is the one when you have some outlier data, parameter of choice to find out the description for a data. So it is not the arithmetic mean. It is one of the positional mean, which is based on their position, right? Its advantage include that it is one of the positional mean, and hence it is not influenced by the extreme values. It is easy to calculate. So the formula that we use to calculate median, there are two formulas. When the data is odd, we have this formula where n is the total number of observation, and when the n is even, when the total number of observation is even, we use this very large and complex formulas, but they are quite simple. So let's see that when your data is odd. So here the number of observations is, in this one, the number of observations is 1, 2, 3, 4, 5, 6, 7. You will have 7 observations, right? So n will be 7. And the median, because it is a small data set, you can easily see that the two halves of the data and the median is in between. Otherwise, you can also calculate it using this formula. The median will be 6. When the data is even, right? So we use this formula. You put the values here and you will get the median for this one, right? The disadvantage of the median is that because it is an average of position, arranging the data in ascending or descending order of becomes time consuming. In case where the magnitude of data is quite large, and also it does not consider the magnitude of the data, it does not consider the actual value of the data, right? It considers only the position of the values. What are these actual values? It does not consider that. This becomes one of the disadvantage for this. Now let's talk about mode in statistics. Mode is simply the most repetitive value or the most frequently occurring value. Whenever you have a particular data derived from an experiment or from any other observation, might find that this data has some repetitive values. Let's say take this example of marks, or where the students have marks out of 10 from a particular paper. So more students or some of the students might have the same marks. Let's say there some students have 3, 3, 3, and 4, 4, 5, 5, 10 also are also there, 2, 3. So this is the marks of the students out of a particular test, or it can be any kind of data. So there are always some values which are repetitive, right? And we have to find the most repetitive value. The most repetitive value will be the mode for that particular data. Let's say in this one, this I think 3 is represented here 3 times. So this 3 will be used as a mode for this particular data set. It is a simple value to calculate and understand. It makes it accessible even for persons with non-mathematics or non-statistical background. It is a robust value which is not by extreme values. When it is not affected by the extreme values, so it becomes quite robust that you can use it in skewed data sets also, right? And it is, in fact, all the measures of central tendency, they can be represented through graphs also or other tools also to compare between two data sets. The mean you can compare between two data sets or three data sets or four data sets. You can also compare the mean between or among different data sets. Similarly, you can compare mode also among different data sets. So let's talk about this. Uh, let's talk about this. Uh, let's say this is our data where we have these many values. So first step in calculating the mode in an individual series is you have to arrange the data in ascending order. So this data is first of all arranged in ascending order. So as soon as you arrange any data in ascending order, it will give you a visual appearance where when the data is small as here, you can quickly find out the mode. Here the most repetitive value is this. 2 is represented 4 times. 6 is also 2 times only. So here this value or 2 becomes the mode of the data. This is the most repetitive value. This is the most repetitive value. But let's say as in this case where we have different values but no value is repetitive, right? No other value is represented even twice. So in such case, there is no mode in the data. Yes, it is possible that the data might not have any mode. And also it is possible that it can have 2 modes or 3 modes also. Whenever we have only one mode, as in this case, we will call this data as unimodal. Otherwise, if there are 2 modes, we will call it as bimodal, and multimodal means there are more than 2 modes. As you can see in this example, the value 3 is represented 3 times. The value 2 is value 6 is represented 2 times, and the value 9 is represented 3 times. So in this case, we have 2 values, this one and this one, 3 and 9. They both are repeated equal, right? So there are 2 modes in this data, which is 3 and 9. One of the disadvantage mode is that it does not consider all data points. It focuses only on the most frequent values and ignores others. Sometimes the data might not have any mode at all, right? So these are some of the disadvantages which are associated with mode. Now, as we have talked about the different measures of central tendency means the single value that is centrally allocated to a data set, and it is going to represent all the values will be called as the central tendency. And we have 3 measures which execute this function. These functions are mean, median, and mode. So is it like we have to calculate all these 3 between in a particular data set? For the purpose of learning, students can or they are made to find out the different values of mean, median, and mode in a particular data set. But let's say you become a statistician. At that time, you might have to decide that which central tendency will be the most useful for a particular data, right? So when you have a symmetric data, right, when you have normal distribution, then the mean, median, mode are always equal. Mean equals to median equals to mode. So in a normal distribution data or a data where everything is symmetrically distributed, you can use any of the value. The mean will be the method of choice to be used. When the data is skewed, it is not normally distributed. The median will be a better way to find a central tendency, because median does not depend more on the values, right? It is a positional average. It depends more on the position of the value rather than its magnitude. And whenever we have categorical data where we have different categories, right? So these categories, when you want to give advantage or when you want to give any visual representation, then mode is the method of choice for using this. So now let's talk about the normal distribution or symmetric distribution of data. So considered the best for any type of statistical analysis is that you should have a symmetric distribution or a normal distribution. A normal distribution data, when it is plotted, will have a bell-shaped curve, right? So here we have this data, and on the lower axis we have standard deviation. So within 2 standard deviation, we should have this 68% of data, 68.2% of data when it falls in within 1 standard deviation, and the 95.4% of the data when it falls within the second standard deviation. This data is considered useful. And whenever you have this kind of data, which is there for most of the cases, uh, for the for the purpose of practice, the mean, median, and mode will have the same value. Your mean will be here, which will be equal to your median, which will be equal to your mode. So all these value will be equal. So this is the data where we have a normal distribution. This distribution may be skewed, and during practical approach, whenever we collect this data, normal data is difficult to find. The data is always going to have values which will be more than the mode, more than the mean, or there will be a particular cluster of data. Like most of the values are in this area, right? And here we have most of the values in this area. So these types of distribution are not normal. They are called skewed, or the data has a property which is called skewness. So these skewed data does not allow to give a true representation of central tendency. So under normal distribution, mean, median, and mode, they are all going to be equal. When the data is negatively skewed, your mode is going to be higher than the median and then followed by mean. While this is opposite in case of a positively skewed data, your mean will be higher as compared to median and mode. So whenever you have to perform any statistical analysis, you have to first see whether your data is normally distributed or it is skewed. So when in under what situations when we can use these measures, like when I should use mean, or when I should use median, or when I should use mode. Whenever you have symmetric data with where the outliers are not there, or if they are even there, their magnitude is comparable to the other values. So near to normal or under normal distribution, the mean is the method of choice to be used. Sorry, the mean is the method of choice to be used when you have normal distribution or near normal distribution of data. Median, being positional average, is beneficial when there are outliers in your data, and it is used in clinical trials, in field trials of crops, literacy data. Median is the most preferred value. Mode is being more related to a category. So whenever you have a categorical data and where you want to identify the most frequent category, let's say you have a census data where you have when you have census data and you want to find out that what is the most the category of an educational class, like 12th pass persons are more, or graduate are more, or masters are more. So mode will be the method of choice. Now let's summarize with this slide, and what are the key takeaways for this. So central tendency, which is a single value that is going to represent the entire data. Three parameters or three measures by which central tendency can be found out in a data set. So these three central tendencies, or the central tendency as a whole, are used to simplify complex data into simple terms, to simplify or make the data more relatable or more comparable across different parameters. Obviously, which parameter you will be using? You will be using mean, median, or mode, depends particularly on the type of data you have, what type of distribution is there. If there are outliers, right, you have to take a data which are not rich in outliers. You should, or even if you collect these outliers, you should remove this from any downstream analysis. At in statistics or in a data set, step for all other downstream understanding of the data. It makes a solid understanding of the data and represent data in a better way, in a simple way. With this, I think we should conclude here the first part.
Yes, sir. We can have a 5 minute break, sir.
Yeah, sure. We can have some break here, and then we will join back. Thank you, everyone, for your time in this session one.
Yes, sir. Before we go, first of all, I want to congratulate you, because when we were assigning this topic to you, we were worried that how such a vast topic will be completed within 45 to 50 minutes. But the way you explained it, extremely commendable. Now I request all the participants to write your name, the same name comment box, and make sure you have to write your name only once. We on typing your name again and again, that will not be taken. So now you can mark your attendance, and after 5 minutes we will assemble here again. Thank you. Okay, so welcome back, participant. I think we can start. Ankur sir, if you are here, you can start. I think Ankur sir is on break.
Yes, yeah. Good evening.
Yeah, good evening, sir. Good evening. Can we start, sir?
Yeah, sure. Okay.
Okay, sir. Just before you start, I request all the participants to mark your queries and question in the chat box, or you can ask your question in the end. Thank you. Over to you, sir.
So the screen is visible.
Perfectly, sir. Perfectly.
Okay. So dear students, and in the second part of this talk, let's move to the other topic, which is introduction to measures of dispersion. So in the first part, we talked about the measures of central tendency. To sum up, central tendency is a value, or it is something that describes a data with a single value, right? But many a times the central tendency is not a method, or it does not represent the actual case. So we will see that what we have to do, or what we have to extend into, right, apart from central tendency, to give more meaning to our data, right? Whenever we have a large data set, we use central tendencies to describe some properties, but these properties are not actual representatives. And when your data is skewed, or in under practical conditions, when you will, right, you say that, let's say, out of 10, 5 is the average. But when we see into this, right, some students will have 1 mark, some students will have 2, some will also have 8, some will also have 9. So this is called dispersion from the central value. This is your central value, mean, right? I am taking the example of mean, but it is also applicable to median, and it is also applicable to mode, right? Your central tendency will have a spread. Not all values will cluster around central tendency. You have a data set where you have 50,000 observations in an actual scenario. So let's say 10,000, 15,000 values will be clustered near to central tendency, but the other values will be far away, and some values will be far, far away. The far, far away values are the outliers, right? So dispersion refers to the spread out of the data. So here we have three data set A, data set B, data set C. So you can see the spread of data set A is less. This is the spread, the area that is being covered. So values are not far away, they are clustered around the mean. In B, these are more far away, and C, the spread is highest. So this dispersion measures the degree of variability or the scatterness in a data set. In very simple terms, we can say that dispersion tells us how consistent or inconsistent the data is. If your spread is less, if your dispersion is less, then the data becomes more acceptable. If your spread is high, if the variability in the data is high, it becomes less acceptable. Let's say I gave example of percentage of students in a particular course in Zakir Hussain College. I said, let's say the students have 80% on an average, right? But if the spread in this is high, some are also having 30%, some are also having 95%, right? So this value doesn't carry the value, right? The spread has to be minimum, right? So whenever we have a central tendency, this central tendency has to be accompanied with a measure of dispersion. What is dispersion? Dispersion is simply the extent of variability or the scatterness or the variability or the spread or the inconsistency in the data. Why dispersion is important? Because the two data set, as I discussed earlier, can have the same average. Right, I'm talking about average or mean here. The same goes with median and also with mode. Two data sets can have the same average but different variability. Right. So let's say here we have this, let's say this one, this data has less variability, right, as compared to this. Right, the mean of both these data set is same, right? Now, which data is more reliable or consistent to be utilized for further downstream inferences? If let's say this is A and this is B, so A is more consistent because it has less spread or less variability. So our data which has less spread is a good data for the purpose of comparing different groups, for the purpose of making inferences about the experiments. So there are a few properties of dispersion that it is always non-negative or its value is going to be more than 0. If all the values are identical, let's say I have a data of marks of students out of 10, right? And all students have same marks. So is there any variability in this? No, there is no variability. All the values are identical. So there is no dispersion. This data has 0 dispersion, and this has no scatter. Right? Or let's say we have this data. So here the spread is less, right? On the other hand, we can have another data where we have values like this. So here we have more variability. So I think this makes a quite good explanation that dispersion represents the variability. It is always going to be 0 or more than 0. If all the data points, if all the observations that you have collected are identical, which is, I think, going to be a very rare case, then the dispersion of the data is going to be 0. Now, we can measure dispersion with two aspects. One is the absolute dispersion or absolute measures of dispersion. These absolute measure of dispersions include range, variance, quartile deviation, standard deviation, and mean deviation. And along with these absolute measures of dispersion, we have their coefficients also. Like for range, we have coefficient of range. For variance, we have coefficient of variance. For standard deviation, we have coefficient of standard deviation. Similarly, for mean deviation, we can have coefficient of dispersion. The one that we studied in the last slide, we have a counterpart, relative measure of dispersion, coefficient of range, coefficient of variance, coefficient of quartile deviation, coefficient of standard deviation, coefficient of mean deviation, right? So for this lecture, I will not be going into the relative measures of dispersion. I will be talking about only these absolute measures of dispersion, because once you understand how to use these absolute measure of dispersion, the relative measure of dispersion is simply an extension of these. Right. So I have written the formulas here for these coefficient of dispersion. So these you can later utilize for calculating coefficient of dispersion for a particular measure of dispersion. Now the first, the foremost and the simplest of all among all the measures of dispersion, range is the simplest one. Its advantage is also that it is also the simplest and it is most easiest to calculate also. Range, right? It tells you the difference from the the smallest value to the largest value. Right? Let's say we have a data set. This data set will definitely have a largest value and it is going to have a smallest value. So the difference between these two range, right? Data can have a smaller range also. It can have a new this data, which is a numerical data here. The smallest value is 3 and the highest value is 9. So the highest value is 9. So we take 9 minus 3. So the range of this data becomes 6. Similarly, in this data set, so this was easy as it is arranged, right? It becomes easy when the data is arranged in ascending order. So the first value will be the smallest, the last value will be the highest, right? But if the data is not arranged, you have to look for the smallest and the largest value. So the smallest value here is 64, the largest value here is 95. So you can take the formula where the maximum value deducted from the minimum value, and so in this way we get 31 as the range for this data set. As I said, it is a simple to understand by even a man of ordinary prudence. It is very easy to calculate. It takes the least possible time to calculate, right? If the data is arranged. If it is not arranged in ascending order, then if the values are more, then you might have to give more time. But if it is arranged, then you have to just look into the first value and the last value. The first value will be the smallest. The last value will be the largest. Its calculation is not affected even if some items in the middle part of the series are missing. So this is one of the merit or great advantage. So in central tendencies, if something is missing, like some values, you were not able to collect data from some time point in an experiment or from a site, so that becomes quite inappropriate. But for range, it doesn't matter. It needs the first and the last value, or the highest or the smallest value. But as it is not taking into consideration all the items of a series, and that is why it is not, it constitutes its demerit also, right? It is giving you a value that depends on the first, highest or the lowest value. It does not consider the data in its entirety. It also does not take into account the frequencies of distribution, right? Like how many times a value is repeated or how many times an observation is occurring, right? It cannot reveal the different dispersal characters of different series in which two extreme values remain same. However, it is good to start with, right? Someone who is new, range is a method of choice. The next one is quartile deviation. So quartile deviation, let's first go into that. What do you mean by quartiles? When we understand what is a quartile, then we can calculate deviation also. Like when we have a data and we can divide this data into 4 equal segments, right? This is the first segment, then this one is the second segment, this one is the third segment. right? So we have first part here of the data, we have second part here, and we have third part here, and we have fourth part here. So the quartiles are going to divide your data into 4 equal parts. So the quartile 1, right? This is quartile 1. This quartile 1 will divide or will have the 25% of the data. In fact, all the quartiles will have 75% of the data. Right. And this one, quartile 2, if you can recall, it is this quartile 2 divides the data into 2 parts. Below this quartile 2, we have 50% data, and above this quartile 2, we have 50% of the data. So this is actually the median of the data. We can call it as quartile 2, and we can also call it as a median of the data, right? And in fact, if we take into consideration only this part, right? So Q1 will be the median for this part, and Q3 will be the median for this part, right? Q2 for the entire data will function as median. Q1 will function as a median for the lower part of the data, and Q3 will function as the median for the upper half of the data. How we can calculate the deviation? The formula is simple. You take value of the quartile 3, deduct the value of the quartile 1, divide it by 2, and you will have your quartile deviation. There is another way, there is another measure which is called interquartile deviation. Interquartile deviation is simply the difference between quartile 3 to quartile 1. So this one that we have is interquartile range, right? So I can rewrite this formula as interquartile range divided by 2 also. This is also correct. Let's understand this by using one example. There is this data. The numericals will be, you have to arrange this data, and this data is already arranged for convenience, right? So if we have to see the range is going to be 56. The highest value is 56 minus 3. So range of this data is 53. Now we have to find out the quartile deviation, right? For quartile deviation, we need to find the value of Q1. We need to find the value of Q3. The formula for Q1 is, and this is when we have odd data, where the total number of observations is odd. Here, the total number of observations is 15. Then we use this formula, where 1 times n plus 1 divided by 4th item, or the 4th observation. This will not give you the value, it will give you the position, right? So here the n is 15, and n is 15 plus 1 divided by 4. So we will have 16 by 4, and the Q1 will become the 4th item. It is not 4 in value, it's 4th observation. So the 4th observation here in the series is 12. So Q1 is 12. Quartile 1 is 12. Similarly, for quartile 3, we have this formula. 3 times n plus 1 divided by 4. So we get, when we solve this one, so the number of observation is 15 plus 1 divided by 4. We will get the 12th item, or the 12th observation. The 12th observation here is 29, right? So we got the Q3 is 29. So now we have to just put these values in the formula. The formula for quartile deviation was Q3 minus Q1 divided by 2. We fill in the values, 29 minus 12 divided by 2, and we get the value. And this is the quartile deviation of the data. This is for when the data is odd. But when the data is even, right? When you have an even series, let's say we have this data, and here the total number of observations is 16, right? To find quartile deviation, right? This is 16. We can divide this data into two halves. We have 8 observations here. We have 8 observation here, right? So we can find median for this data and we can find median for this data, right? Let's take this numerical. So here I am taking this formula and putting in the values. So the total number of observations is 8 divided by 2 observation plus, here we have 8 divided by 2 plus 1 observation and divided by 2. So when we solve this one, right? When we solve this one, we will get the values for this one will give you the value for quartile 1. This one will give you the value for quartile 1. And this one the value for quartile 3. I'm not going to solve this entirely. You can use this formula, put in the values, you will get the values for quartile 1 and quartile 2, right? You can also, because it's a small data, you can also see that the 4 values are here and the quartile will be someone here between these positions, right? And it will be here 4 values here up on the lower side and here up on the lower side and 4 values on the upper side. I think it is now clear to you that it is easy to calculate with the formula in the odd series. For the even series, you can use the formula of the median. What are the merits and demerits for this quartile deviation? Obviously, it is easy to calculate and understand, is far much better than range because it considers 50% of the data. The range takes into consideration only the first and the last value when the data is arranged, or the highest and the smallest value, right? And the demerit is that because the same is also demerit. It considers 50% of the data for the calculation, while it also ignores 50% of the observations. Some of the observations, or 50% is quite a good number. If you ignore 50% of the data from a calculation, then obviously that is not a good measure. The values that are coming as the answer are not true representative of the entire data. It does not exhibit any scatter around an average, for which is remarked as measure of partition rather than a measure of dispersion. Also, it takes more time to calculate. It is mainly in comparison to range. Obviously, if the method is more reliable and robust, like mean deviation or standard deviation, they are going to take more time, right? So we cannot, in today's time, we have many tools. In fact, even with the help of Excel, which is available on laptops and desktops, Microsoft Excel, this becomes a task of seconds. I think in the coming lectures, you will have the idea of how to use different tools and how to use Excel also. Excel will be the very basic.
Yes, sir.
Right. So now let's come to mean deviation. So whenever I'm saying deviation, I think you should be clear about this. Whenever we have a measure of central tendency, all the data, we cannot assume that all the data will be clustered around that mean. Let's say we have this mean value. So all the values will not be clustered around this, right? Some values will be far away from the mean. So this is called deviation. This point is deviating from the mean. This is even more deviation, and this one is even more deviation. So deviation can be in both directions. Mean deviation, which is also known as mean absolute deviation, it is the average of absolute differences between each data point and the mean. It tells us how far each data is from the center point of the data. The mean deviation is represented or can be calculated using this formula. This is the sign of summation. This is going to be your observation. You will fill in the value of your observation of each point, and this is the mean of the data set. N will be your total number of observations. Right. So, right. Why mean deviations should be used, or any measure of dispersion should be used. So I have this data here where we have three data set 1, data set 2, and data set 3, right? So the mean of all these data is same. It is coming 50, right? So it looks like that the values which are there inside these data sets should be also similar, right? But it is not so. Just a minute. Right, when we see into this, the values are quite different. In the data set 1, we have huge deviation, right? One value is quite large as compared to other observations, right? This one is, I think, is the most symmetric data. This one is moderately symmetric, and this one is like a vague data that cannot be used. If you are doing your analysis using this data set 1, then it is not going to help you to get meaningful result. Now let's see one example here. So here there are different families which are alphabetically named. Family A, B, C, D, E, F, G, H, I, and J, and that how many siblings are there. The number of brothers and sisters is being asked, right? So some families have, A has 3 siblings, B has 2 siblings, family C has 2 siblings, right? So this is the data that we have obtained. So when we take mean of this data, how can we take mean of this data? What we have to do, we have to take the total number of observations, summation x, which is 29, right? Summation x divided by total number of observation, which is 10. So we do this 29 divided by 10, it is 2.9. So almost 3 siblings should be there. This mean or the central tendency is telling us that there are 3 siblings per family. But as we can say, we have 1 sibling also, we have 2 also, we have 2 also, we have 4 also, we have 6 also. So these are called the deviations from the mean. We have mean and then we have deviations from this mean. Now we are going to subtract each observation. The observations are these. These are the observations. We are going to subtract these observations from the mean, like x minus x bar. It is 3 minus 2.9, right? So we will get 0.1. Similarly, we subtract each value, each observation from the mean. 2 minus 2.9. So we get minus 0.9 and we get this data. In mean deviation, we ignore the signs, right? These negative values, these negative signs, we will ignore and we get this data here, which is referred as dx, right? The deviation from x. And when we take a sum of all these deviations, we add up all these values, which is coming to be 9.2. So the mean deviation is of this data is 9.2 or 0.92. So almost whenever we have to tell someone that what is the number of siblings per family, in this particular locality, we will say the mean is 2.9, but it has a mean deviation of 0.92, right? This is more robust way to explain any data, right? We have to utilize central tendency along with the measure of dispersion. Now we have standard deviation along with variance. Standard deviation and variance are related, and they are, in fact, the standard deviation is considered as the most widely used and the most reliable parameter of deviation, right? That is why it is called as the gold standard. What do we mean by gold standard? Which is used most, right? So this standard deviation is represented by this sign rho, or sometimes also written as SD, standard deviation. It quantifies the average distance from each data point from the mean, right? Most commonly and reliable used measure of dispersion, providing a comprehensive understanding of data spread. Data can have a smaller standard data, might also have a large standard deviation, which means that values or the observations that you have collected are more spread out from the mean. So from the bell curve, right, this is normal distribution spread, right? The spread of the or the variability is more, right? So this data has high standard deviation. While if your data, when you plot your data, looks like this means that you standard deviation, right? So which data is more useful? Which data is more reliable? Which data is going to produce more consistent result? This one, this data, where the standard deviation is low. So whenever you collect our data, you should see that how much standard deviation is there in this one, right? It should be not like this, that you have collected a data and you moved forward by through analyzing various methods of inferential statistics like chi-square test or t-test or any other test that you are going to perform. Data is good enough for downstream analysis, right? And how will you make sure that your data is good enough? It should be utilized for downstream analysis or not? The one parameter that you can use to make sure standard deviation, and it is a common opinion about among statisticians that your data should have or should not have more than 2 standard deviation. If your data has more standard deviation, like it is widely spread, it should not be used, right, for downstream analysis. It should have less spread, and the spread should be equal to 2 standard deviations, right? So it depends on the type of data also, but for a ideal condition, you should have a low standard deviation. So the formula that is used to calculate standard deviation is this, where you have sign of summation x minus x bar divided by n minus 1, right? So this n minus 1 is used when you have less than 30 values. When you have less than 30 observations, we use n minus 1. This is called as Bessel's correction. There was a person who otherwise the formula is just you have to be right the total number of observations, but Bessel suggested that if you have less number of because standard deviation is calculated mostly on samples, right, not on entire populations, when I say population, it is not a term of ecology. In population, we mean the entire set of data, the original set of data from which we are going to collect samples. Right? Let's say we have a very large forest. We have a very large forest, and this forest has 50,000 trees, and we have to find the width of these trees, right? So we will not be going to find elements. We are going to pick samples from different locations at random through random sampling methods, right? And these samples, if they are less than 30 in number, we should use n minus 1. If they are more than 30, we should use n only. And in fact, this one, the formula which is under this, right? If we talk about only this one, this part, this is referred as variance. So I can also say standard deviation is the under root of variance. Yeah, it is written here. So the formula is summation under root of summation x minus x bar divided by n minus 1 if your sample size is 30 or less than 30. If it is more than 30, we will not use n minus 1. We will use only n. And this thing that this part only, which is under the under root, is variance, right? So standard deviation equals to under root of variance. And as I said, then you have a normal distributed data. This data should be under 2 standard deviations, right? Your entire data should, or your spread should fall in this. If you have more deviation leading to -3, -4, -5, right? And here also, then it is not fit for downstream analysis. Right, now let's take one example to have a better understanding of standard deviation. Right, so let's say we have 2 plants. These plants are, let's say, hybrids or transgenics. These plants are, let's say, hybrids or they are transgenics, right? Or and the scientist is saying that these plants yield more seed per number of plant, right? So we collected the scientist carried out a field trial, and this field trial we collected samples or the seeds from different plants. So I think there are 10 values, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. From 10 plants were randomly selected, and the seeds per plant are counted and their observations are listed there. There are seeds per plant. So in plant A, which the scientist is saying is more higher yielding, let's say it's a rice plant, and this is a rice plant B, and one other scientist is high yielding, right? So how we can say that which one is more high yielding variety? So one way is to find out the mean, right? What is the mean or average number of seeds per plant? For finding the average number of seeds, we have to take into consideration the total of this, which is 240, right? And total of this, which is 100. So for finding the mean, we have total of all the observations divided by number of observation. So it becomes 24. 24 seeds per plant, right? Similarly, for second plant, we have 10 number of seeds per plant, right? We have 10 number of seeds per plant. So it seems like that this plant A or the variety A has more number of seeds per plant. But as I said, the central tendency, which is mean in this case, is not a true representative of data. We have to also consider the spread. Now to see the spread, here we have represented X whenever we have more data, right? So we can write X my bar 1. This means it is the mean of dataset 1. This is the mean of dataset 2, right? This is the mean of dataset 3. Similarly, we can write standard deviation for dataset 1, standard deviation for dataset 2, right? Variance for dataset 1, variance for dataset 2. right? So here we are going to do the same thing as mean deviation. We are going to find out, we are going to subtract the mean from the each observation, and later on we are going to square this up, right? We are going to square this. So 4 times 4 becomes 16, and the negative sign will go away, right? And then we will take a summation of this. We will sum up this data, this one, right? It is coming 136, right? And similar procedure will be done for the data set 2. We will find the deviation, right? And then we will take the summation of these squared. So we will fill in these values in the formula. So this summation of x minus x bar for data set 1 is 36. And why we have 9 here? Because n minus 1. So total number of samples were 10. Minus 1 becomes 9. So it becomes 136 by 9. So the variance for dataset 1 is 15.11. And similarly, the variance for dataset B becomes 21.33. And for finding the standard deviation, we have to take the under root of variance. So the standard deviation for these two becomes 3.887 and 4.619. So now we know that plant A or the variety A is giving more seeds per plant, 24, and also the spread is less. The consistency is more. So this should be given a go ahead. Now let's move to variance. Variance is the average of the, we have already seen this in the previous slide, but to be covered as a separate topic, variance is the average of the square differences from the mean. It penalizes larger deviations more. The key difference is the absence of the square root from the standard deviation. The key difference is the absence of square root, which means variance is in squared units. Less intuitive for direct interpretation, but essential for mathematical properties. Variance provides the raw measure of spread, which from which standard deviation gives a more interpretable value in original units. Variance you can also calculate from standard deviation. The square of standard deviation will be your variance. So which parameter you should use? You should use, if you have a data, standard deviation and variance should be used. Now, key takeaways from this part of session, that measures of dispersion, right? Or we say that whenever we have collected a data, what suits with your data is depending on the requirement of the experiment. And as an informed person, as an informed statistician, you should have a proper understanding of these before taking any decision, right? In today's time, the tools are going to give you suggestions, but that this analysis or this parameter should be used, right? But for starting or for making a foundation, that you should be aware of the merits and demerits of all these data, or all these parameters, and you should be aware that how your data was collected, whether it is normally distributed or skewed, whether downstream analysis in this is required or not, or what outcome you want from your data. Based on all these considerations, you should be take an informed decision that which parameter of central tendency or which parameter of dispersion suits you for your best informed for your outcome. I think with this, I thank you all for your time and your patience on this Saturday evening. right? And your colleges must have started from yesterday, and but you all have taken time to come out for this kind of educational series. That's a commendable job, and I'm also thankful to the team of this uh, workshop and the principal of Zakir Hussain College, all the members for giving me this opportunity. Over to you, Dr. Savin.
Thank you very much, sir, for your time and your presentation, which was truly engaging and thought-provoking. Now we encourage all the participants to take this opportunity to ask any question or share your queries with Ankur sir. Please let us know. You can type your question in the message box, chat box, or you can directly raise your hand and ask the question. Although there are some questions at my end, I have received some of them. Yeah.
Yeah. Meanwhile, the students are generally if you are when they are thinking, let me give them if we still have 2-3 minutes. So can I give one practical scenario where statistics plays a role? Because this
Yes.
is what we have
Yes, yes,
talked
yes.
about is quite theoretical, right?
Yeah.
And we these students might have been studying about mean, mode, median. Dispersion might be new to them, but how you can use them. And because I am a student of biological sciences, and I worked on large data sets. Actually, I had used tools where the tools recommend that what type of analysis that you should perform. The tools are quite advanced these days. But for a starting point, let's say that when we uh, do a real-time PCR and we get an expression of a gene, right? So what are the chances that when I am going to repeat this experiment, the same gene expression will come, right? And even if it is coming more or less equal, that what are the chances that it will come same as the third time? Because in biological sciences we repeat the experiment, same experiment 3 times to see that what if the results are reproducible. But nowadays people say that your results should be accompanied with statistical analysis, like a t-test should be done there, right, to give more consistency to your data. And also you should place error bars on the expression values, that your values you cannot say just that my expression is twofold, it might change in other experiments, right? So you should also place error bars on your results. So these things are required in today's time. Whenever you communicate a paper, a research paper, or an article, people, and in fact, it gives a like this salt to your entire preparation. Then you have a good experiment and you add some statistical analysis, it becomes the complete dessert, right, for the reviewer.
Thank you, sir, for this real-time example. Now I have received two questions. So one is the one student is asking if a data set contains a large standard deviation, can we reach the right conclusion from this analysis?
When the data have very large,
Large standard deviation.
it is preferred that you should have a smaller standard deviation. But in cases where it is not possible to have a data with a smaller this thing, then you have no choice to proceed with this. But obviously, your results, you have to supplement your results with other tools also, right? You cannot just rely on standard deviation or something like mean or this thing. Yeah, it is said that that your data is good if you have a smaller, but if you have no ways to generate a data where the standard deviation is quite large, so as the value of standard deviation increases, your data becomes less and less fit for downstream analysis.
Yes, like nowadays, I think there are some software which can also remove some outlier.
Yes,
So
yes.
if we will remove the outliers, obviously the standard deviation will be lower.
It will come down.
Yeah,
Yes.
there are similar kind of question. Can we use skewed data for inferential statistics? I think the meaning is same.
Uh,
So if there is any, I think there is no more question. So thank you very much, sir, for your time. Now it's time for me to formally thank through the vote of thanks. Sir, some students are asking, can you share your presentation? Will it be possible for you?
yes, yes, I will send it to you. Then you
Okay,
can
sir.
share it with your.
Otherwise, they can directly use the YouTube because the entire lecture is being uploaded on our college YouTube
So,
channel.
yeah.
So all the slides are readily available with you. So now, on behalf of the organizing committee, I take this opportunity to express my heartfelt gratitude to all those who made this event a success, and especially on the weekend, Saturday evening, when everyone have a separate plan, but because the colleges are open, that's why we have to shift our timing from 11 to 1 to in the evening. First and foremost, our sincere thanks to Dr. Ankur Bhardwaj for gracing this occasion with your presence and sharing such valuable insight and concise this a big and vast topic. Each and every topic, like your entire lecture is divided into two parts: measures of central tendency and measures of dispersion. And usually we take 3 4 class when we are taking in our colleges, but you are able to complete both the topic within 1.5 hours. So a huge round of applause should be there for your effort. I extend our deep appreciation to our principal sir, Professor Narendra Singh, whose guidance and constant support made this program possible. To the IQAC coordinator, Professor P.K. Sisodia, and the bursar of the college, Professor Mukesh Jain, thank you for your constant support and guidance. I also acknowledge the constant effort of the convener SDCC, Dr. Saima, and fellow colleagues, Professor Dheeraj Singh, who ensured the smooth conduct of the session. A very special thanks to all the participants who have registered and shown such enthusiastic engagement. Your presence is what gives us meaning to this course. Last but not the least, thank you all once again for being with us today. We look forward to continue learning, collaboration, and success throughout the course. Now you all can mark your attendance in the chat box, and it's a gentle reminder to finish the last week assignment so that we can release the answer and as well as score. Last week we were unable to release your score because of some technical error, but this time you will also go to know not only the answer as well as your how you have performed in your last week assignment. So make sure that you will finish it by today midnight. And now it's time to say bye bye everyone. Have a nice weekend, and we'll see you tomorrow around just before 11 a.m. And we will have Dr. Sweta Sharma from Maitri College, who will actually present our data through graphic. Thank you everyone.